I'm Lex van Geen. I'm the Associate Director of the Superfund program. I'm a geochemist here at Lamont Doherty. And I'm happy to be standing here next to Mason Stahl. Mason studied math and economics as an undergraduate at Tufts. And I don't know if there was a gap, but then started working with Charlie Harvey at MIT. And so, someone said switch fields and has done a fantastic job there. We'll hear about some of the work that he's conducted, primarily with Charlie. But I also want to tell you that Mason did a number of things with us during those years as well. He wrote the first paper demonstrating that you could test for leaks in a well in Bangladesh by spiking the water with salt and coming back the next day to see how the salt was distributed. And that was, we discovered that some of our deep wells were high arsenic because they were badly constructed. Mason was also very much involved in the study that we conducted, and with Baltic played a big role in that in Vietnam, where we showed that the front of high arsenic was moving from one low arsenic aquifer to forward about 20 times more slowly than the water was. Mason did a lot of the analysis involved with that. And but today is going to talk about something else, a topic that I think we as a group here will continue to study, and hope we continue to study, which is what is the ultimate trigger of this arsenic release to groundwater. And you will see, Mason did a type of field experiment of a scale that we certainly haven't reached, but is very interesting for that. So Mason, oh, maybe I should have mentioned, I'm sorry, so Mason defended his PhD a year ago, something like that, and is now working at Radiant, which is the same sort of high-end consulting company where a former graduate from our program, Terry Redox, is working today.
Thanks so much, Lex, for the introduction. So let me just pull up the slides. Oops. Oh, right there. Great, thanks so much. Yeah, thank you so much for having me here today. So I'm going to be talking about some work related to groundwater arsenic contamination in South and Southeast Asia. I want to thank, first off, the funding agencies, NSF and NIH, as well as my collaborators, so my advisor, Charlie Harvey at MIT, and our two collaborators at Bangladesh University of Engineering and Technology, Professor Borhan Badruzaman and Mehdi Hassan Tarak. All of them were instrumental in this work. So I'm going to be talking about a study I did looking at carbon dynamics and redox changes in an arsenic-contaminated aquifer in Bangladesh. So first I'm going to present and discuss some background on the issue of groundwater arsenic contamination in South and Southeast Asia. Then I'll discuss a few questions that motivated my research and the relevance of these questions to the broader issue of groundwater arsenic contamination. I'll present the work I did at my Bangladesh study site. We're at a multi-year field experiment looking into these questions, and also some laboratory experiments that I ran. I'll discuss the results from this field and laboratory experiments and the implications of our findings towards the broader issues in groundwater arsenic contamination. I'll end off with some concluding remarks and a brief discussion of future work. So I'm sure everyone in the audience here is very familiar with the fact that arsenic is highly toxic to human beings. So if you're ingesting groundwater with high levels of arsenic, it can result in a whole host of health issues. So increased incidences of cancer, cardiovascular disease, diabetes, and cognitive impairment. So it's really problematic. And the issue in South and Southeast Asia is that many of the shallow and superficial aquifers are highly contaminated. So the groundwater is highly contaminated with naturally occurring arsenic. So if we look at the country of Bangladesh, it's estimated that 50 million individuals are consuming groundwater, are exposed to groundwater that exceeds the WHO guidelines of 10 parts per billion arsenic. And now this problem is by no means confined to Bangladesh. So if we move eastward and look in Vietnam here, it's more than 1.5 million individuals. If we look in Cambodia, it's more than 500,000 individuals. And if we look in the state of West Bengal, India, it's 30 million people. So this is a widespread problem affecting tens of millions of people. And since the problem came to light several decades ago, there's been a large amount of scientific investigation to try to understand what's causing this natural pollution. And we've learned a lot in that time, but there are still some open questions. Some of these questions motivate my research here today. One thing we do know well is the general mechanism responsible for arsenic mobilization in these aquifers. So this mechanism is the reductive dissolution of arsenic-bearing iron oxides. And it's a microbially driven process. And right here, I'm showing a cartoon of what goes on. So in all aquifers, you'll have organic carbon. So think of it as plant matter that either enters with recharging water or is deposited along with the sediments. And then you'll have microbes. So that's indicated here. That little guy is a microbe. And those are in all aquifers as well. And then there's arsenic-bearing iron oxides. So in the sediments, you'll have. Oh yeah. So then you have the arsenic-bearing iron oxides here. And these, so one point about iron oxides is that they have a strong affinity for arsenic. So arsenic can get sorbed to the surface or incorporated into the mineral phase. And if it's in the solid phase, it doesn't pose a huge risk to people. But when it becomes released into the dissolved phase, that's where the issue of groundwater arsenic contamination happens. And this is driven by microbial reduction of these iron oxides. So similar to humans, microbes need to eat a source of organic carbon. They need to build biomass and get energy from this. And when they eat that, they need to breathe something. So we would breathe oxygen, but in an aquifer, you're not exposed to oxygen. You're out of contact with the atmosphere. So they've evolved to breathe iron 3+. So this is oxidized iron. They'll reduce the iron 3+, and then it becomes soluble, goes into solution, breaks apart the mineral, and can release any associated arsenic. And as they do that, it's an important point is that when you oxidize organic carbon, you're going to generate CO2. So we respire CO2, same with microbes. And this becomes dissolved in organic carbon. So that's the general process responsible here. And so to summarize here, we know that arsenic is mobilized through the reductive dissolution of iron oxides, but we don't really understand how current levels of dissolved arsenic reach their present state. And in particular, we need to better characterize the organic carbon sources that are driving these reactions. So we've asked the question of what are the organic carbon sources driving arsenic mobilization? Are they surface-derived sources of organic carbon? So is this organic carbon that maybe is coming in with recharging water from ponds, wetlands, rice fields, or rivers? Or is it organic carbon that was co-deposited within the aquifer? So organic carbon that deposited thousands of years ago. Now, this is an important question, and there's been lots of research into the issue, and it's pointed in both directions. So we really wanted to resolve this. And it's particularly important because you can think about two end-member cases. So I just want to highlight why we really care here. Imagine if we find out that it's just surface-derived organic carbon that's driving these reactions. So arsenic is being mobilized through the reductive dissolution of iron oxides fueled by surface-derived organic carbon. Then that could suggest that you might have high arsenic. The generation of high arsenic may only be possible if you have surface-derived inputs, because there's already sedimentary organic carbon there. And if we find out if it's surface-derived driving it, it suggests that you might need surface-derived organic carbon to drive these reactions. And then that has important implications. It says that if we change the delivery of these surface sources, we might affect arsenic concentrations. So the change in delivery could be due to the creation of wetlands or ponds or the draining of them. And now what if we found out that it was just sediment derived? This is the other end member case. So if we find out that it's just sedimentary derived, that organic carbon that's driving these reactions, then we can say that, then we might think that high arsenic is, or we would know that high arsenic is possible without the input of surface-derived organic carbon. So this is sufficient, not necessarily necessary, but it is sufficient, and that's why we have high arsenic. And I just want to point out, if we do find this, it does not rule out the possibility of the surface-derived organic carbon driving the reactions if more was introduced. It would just suggest that the present state that we see is driven by sedimentary organic carbon driving these reactions. So our overall research goal here is to improve our understanding of the organic carbon sources driving arsenic mobilization. Now, this is a relevant question. I just pointed out some of the relevance here, but more broadly speaking, if we better understand the organic carbon driving these reactions, then we'll understand how aquifers are going to respond to perturbations. So how will an aquifer's arsenic and organic carbon cycle respond to a perturbation such as land use changes? So if we convert, say, urban land to farmland or farmland to urban land, if we build or drain wetlands, things like that. And also things like intensive groundwater pumping. So throughout South and Southeast Asia, there's intensive groundwater pumping for urban water supply and for irrigation of crops, in particular rice. And then you could have climatic and hydrologic shifts that might also perturb the system. Furthermore, when we better understand the organic carbon sources driving these reactions, we'll be able to understand what arsenic concentrations may have looked like in the past, why they look the way they do in the present, and then how they're going to respond and change in the future. So now that I've established the questions driving my research, let's move over to my field site in Munchiganj, Bangladesh, where I worked and had my field and laboratory experiments to answer these questions. So Munchiganj, Bangladesh is about 30 minutes south of the capital, Bangladesh, Dhaka. And here I'm showing a picture of a study pond that we constructed to help get at these questions. Now I'm going to highlight in the next few slides why ponds are important and why we care about them. But basically, they're a surface source of organic carbon that could be entering the aquifer. So let's look at a satellite image now of my field site. So I've highlighted the ponds in blue to make them stand out easier. They're kind of hard to see on a satellite image. The other areas are rice fields, and then you have villages here, the raised villages, and those are shown, they look like there's trees there, and so those are all the treed areas, those are villages. So you've got mainly ponds, rice fields, and villages. And the reason there are so many man-made ponds in Bangladesh, the vast majority of these ponds being man-made, is because Bangladesh floods annually. So there's monsoonal flooding, and it can cover a large proportion of the country for a time. And if you build your house on the land surface and it floods every year, your house is going to flood every year. So you want to avoid that. So what people have done, and throughout time there, is they've excavated sediments. They build mounds, and then they put their villages on top of these excavated mounds. So these mounds are several meters above the land surface. Okay, well, when you excavate all that sediment, now you've got a hole in the ground, and that becomes a pond. It'll fill with monsoon floodwaters. That floodwater is retained and can recharge the aquifer. So these ponds aren't just kind of a pointless result of building villages. They play an important cultural and economic role. Historically, they were used for water supply, waste disposal. They're used for bathing, and aquaculture, so people will farm fish in them. So we know that these ponds are ubiquitous, and they can provide a source of organic carbon-rich recharge to the aquifer. And this could stimulate both arsenic mobilization in the near zone, in the shallow zone, or it could carry labile, very reactive organic carbon, into the deeper zones in the aquifer and mobilize arsenic there. And this recharge can be carried faster because of your intensive irrigation pumping. So there's lots of irrigation pumping, and it can rapidly pull this water down into the aquifer. And prior research has suggested that ponds may be playing a role by introducing this reactive organic carbon and stimulating arsenic mobilization, and even potentially generating plumes of high arsenic coming out of the bottom of these ponds. So we wanted to understand what are the relative roles of ponds, and also just surface sources more generally, versus sedimentary organic carbon sources in driving these reactions that are responsible for arsenic mobilization in Bangladesh. So our approach here was to construct a study pond, and study both the organic carbon cycling and the redox transformations that are happening beneath this pond, as well as throughout the aquifer at our field site. Okay, so like I said, we constructed our own study pond, and the reason why we started with a brand new pond is because if we had taken a pond that's 50 years old and we studied the geochemistry beneath it, it'd be very hard to tease out what was due to the pond and what were just the prevailing conditions. So by starting with a fresh pond, building a brand new pond, we could see these transformations happening over time, and we could tie down and say, oh, okay, that's because of the pond. These geochemical shifts are due to the pond. So here's a picture of our field site. And this is a rice field, and this is in May 2011. So right after the rice was harvested, we started construction of the pond. So now you're looking at our pond under construction. So here's a picture. I'm in the middle here, and this is the team that we hired to build our pond. So this is following local techniques. These ponds are commonly built. You'll hire a team of workers. They'll come, they'll chop away the superficial clay. They'll carry the clay on baskets, and they'll build a berm around the pond, and they can use any excess clay to build up a village. So here's our pond under construction, and then this is our team here, continuing to build the pond. It's taking shape, and now this is a few weeks into the construction. You see that the pond's really taking shape. We've got the depression here and the berm coming around the pond, and we started to build a dock in the center of the pond so that we could put instruments there and sample the pond from that dock. Now here's the pond after it's completed. So this is before the flood season. Our pond is finished. It's not really full of water yet. A little groundwater seeped in. But you see the berm is around it. And then you'll also notice all these pipes sticking out of the ground. So this is a network of wells that we installed. These are monitoring wells, and this would allow us to both track the geochemical shifts and the hydrologic shifts around the pond. So they span the whole depth range through the aquifer. And we also had a dock in the center where we installed clusters of wells going into the aquifer, but from the center of the pond. And that way we could track this vertical recharging plume. And then we had shallow soil water sampling. So, like I said, the field site floods every single year, and much of Bangladesh does, in fact. So this is our pond flooded now. I just want to flip back a slide. So you see the berm here, and the berm here in the top in the right of the picture, that's several meters above the land surface. And now when we look back at the flooding, it's just, maybe a foot below that berm. So this is under like 6 or 7 feet of water at our field site. So it's flooded, it's filled our pond with water, and you see our dock here. And then now the floods receded. So the floods receded, and our pond has still retained a lot of water because we have this berm around it. And now the pond can recharge, and you can tell that it's probably organic carbon rich. There's all this plant material, and if I had a closer picture, you'd see like kind of algae and scum growing on the pond. So this is a very organic carbon rich source of water. Okay, so we first needed to characterize the hydrology. And this talk is about geochemistry, so I'm just going to give you kind of the takeaway point here. But we did a full characterization of the water balance and the hydrogeology of our site. But really the main point I want you to see is that we have a large downward flux labeled G here. So this is the flux from the pond down into the aquifer. And this is going to carry that water that has organic carbon. And the flux is driven by a hydraulic gradient. So you can see that the water level in the pond is above that of the water level in the underlying aquifer. So if you look at the well, in this picture, we've got a well screened in the aquifer below the pond. The water level is lower in that well. So now there's a gradient, and it drives this water into the aquifer. Okay, so we did our hydrologic characterization. We also did an intensive geochemical characterization of our field site. So I just want to show you the map of all of the sampling points that we had to conduct our geochemical characterization. So each of these black dots represents an individual well, and you'll see they're in groups of 8 mostly. So there's a cluster of wells, and at each location we'd span the depths to go down from the shallow surface down into the aquifer to the depth of arsenic, the highest arsenic, which also coincides with irrigation pumping depth. And then we also have some of these other clusters here, these squares. These indicate groups of shallow soil water samplers that we installed. Okay, so I just want to show you now the field site in cross section so you understand the geometry and you can see how we're doing our sampling. And what you can see here is that we're really covering we're spanning the whole depth of the aquifer from the shallow zone down to the depth of irrigation and high arsenic. And we really want to understand what's going on, why we see the arsenic concentrations that we see today and why we see these patterns and what the organic carbon source is driving this is. So let's take a look at these arsenic concentrations within our aquifer. Right here, I'm showing a figure that shows arsenic versus depth. Each of the gray circles represents an individual measurement on a well, and then the blue squares represent the average concentration at that depth. There's a few things you can note. You see the shallowest wells at around 5 meters. Those are near the WHO guideline, but they're still above it. And then we have arsenic concentrations increasing with depth, and they become incredibly elevated once we hit 30 meters. This is upwards of 100 times the WHO guideline for safe drinking water. So we have really high arsenic levels there. So one thing we want to understand is arsenic being mobilized along flow paths as we move down? Or is arsenic being mobilized, it's very shallow and then focused at depth? So maybe we're having a plume of high arsenic generated beneath a pond or a wetland, and then it gets focused at depth. Or potentially some combination of the two. So this is something we wanted to look at. Well, first we should take a look at the shallow arsenic concentrations beneath our pond. Maybe we could see high arsenic there. So let's zoom in here now and look at the geochemistry that's occurring beneath our pond. And we're going to look at the shallow pore water samplers beneath our pond. We can ask this question, is arsenic released in the near surface during recharge? So now I've added the points. If you just look at the top of this figure, I've added the points from our shallow samplers. And basically what you can see is that those concentrations aren't very high. There are individual measurements, gray circles. They're actually just covered by the mean values here because there's not a huge range of variability. So you see that we don't have a high arsenic at shallow. So that suggests, at the very least, our study pond and in the area around our pond, there has not been a plume of high arsenic generated in the shallow zone. but we definitely have high arsenic at depth. So we need to start investigating further. And the way to do that is to look at the carbon balance beneath our pond, as well as throughout the aquifer. So what we did was we investigated the carbon cycle beneath our pond to determine a few things. One is to determine the organic carbon source that's driving these redox processes. Remember, this is the mobilization of arsenic is a redox process. You're going to oxidize organic carbon and reduce iron oxides. Oh, sorry, yeah. It's actually kind of tricky. Okay, so you're going to reduce iron oxides and then release arsenic into solution. Okay, so we want to identify the organic carbon driving these redox processes, and then also see, is this organic carbon being carried to depth? So maybe it doesn't mobilize arsenic at shallow, but you could have really reactive surface organic carbon that gets carried into depth and can drive the mobilization of arsenic deeper, and we definitely know that that's happening because we see high arsenic. So before I move forward, I just want to give a quick overview on carbon and the different forms it could take, because this is going to be important in understanding the next upcoming slides. So there are two types of carbon you can see. There's organic and there's inorganic. So I'm showing organic in red, and then I'm showing the inorganic carbon in blue. And then it can be in two phases we can think about. We can have solid, so these are shown as squares. Just kind of easy way to remember it, just think of like this, these are squares kind of like a brick, so a solid phase. And then we have dissolved, these are shown as circles. Now I'm going to step through the four types we have here and describe them. So you've got organic carbon in the solid phase. You can think of this as plant matter or bacteria that this could have entered and been deposited with the aquifer. And then you can have dissolved organic carbon, right? And this is just now a functional cutoff. We say that it's dissolved if it can pass through a 0.2 micron filter. And then you have inorganic carbon. And I'm sure you're all familiar with this. These are carbonate minerals like limestone. So calcium carbonate is a solid phase of inorganic carbon. And then dissolved inorganic carbon is CO2 that gets dissolved in water, and it can form carbonic acid, and it can take on a proton or give up a proton. And these are the different forms there. So that's shown as the blue circle. And what's really important here is the transformation, so going from organic to inorganic. And that's going to be play an important role in the upcoming discussion here. So now this diagram here is just showing what goes on and how you can move from one type of carbon to another type of carbon. So you can go from solid to dissolved through a dissolution reaction, but really what's important here is going from the red to the blue. We really care about going from organic carbon to inorganic carbon because that gives us a proxy or an indicator of redox reactions. Remember, you're going to oxidize this organic carbon, and you're going to couple that to the reduction of something like iron oxides. So if we track the inorganic carbon that's generated, then we can understand what the organic carbon source that generated that was. And this is going to be driven through consumption microbial processes. So the microbes can consume solid organic carbon or dissolved organic carbon and generate DIC. So that's really the important process, organic carbon to dissolved inorganic carbon. Okay, so now let's return to what's going on beneath my pond. We looked at the arsenic concentrations beneath it. Now let's investigate the carbon cycle. Just to remind you, we want to determine these organic carbon sources driving the processes, and then identify if these organic carbon sources can be carried to depth and continue to drive processes within the aquifer here. Okay, so this figure here is now showing some carbon samples that I've taken from beneath my pond. Now these are all dissolved carbon. So organic is shown in red, inorganic is shown in blue. And there's you don't need to get lost in the data, but there's a couple takeaway points that I'm going to highlight here. So if we look in the pond water, and so the shallowest sample there, that's the actual surface pond water. And then you go to the shallowest sample in the sediments, you see there's a huge increase in dissolved inorganic carbon, right? And remember, dissolved inorganic carbon is generated when we oxidize organic carbon. So and couple that to a redox reaction. So there's redox processes going on, and we're generating a lot of DIC. But if you also look, you'll see that the change in dissolved inorganic carbon as you go from the pond water and into the shallow sediments, that change in concentration is much larger than the concentration of dissolved organic carbon. And this conversion is a one-to-one. Like 1 unit of organic carbon, when it gets oxidized, goes to 1 unit of inorganic carbon. So what does that tell us? Well, it tells us that the DIC increase is bigger than the DOC that we're measuring. So there must be a solid phase source of organic carbon, and this is leaching into solution, getting oxidized quickly, and keeping the organic carbon concentration low, but generating large concentrations of DIC. But furthermore, we saw that there's a lot of DIC generated in these upper 50 centimeters of sediment. So that means that we have vigorous redox reactions going on here, but we did not see high arsenic. So there's something fueling these reactions, but but we have yet to generate high arsenic here. But that does not mean that we couldn't bring this reactive organic carbon to death and start fueling the arsenic mobilization there. Yeah, so we did that, and I'll show on the next slides. We used carbonages, and there's very low concentrations of calcium carbonate. So from mass balance calculations, it couldn't have been supporting this. That's a great question.
Sorry, when you say these reactive organic carbon, what do you mean by that?
So that it was consumed and goes from organic carbon to dissolved inorganic carbons.
Are you meaning that from the sediment or part of the DOC?
Yeah, so the question was, what is the reactive organic carbon that's generating the DIC? Is that?
Right.
Yeah. Okay. Well, I'm gonna get to that on the next slide or two, but yeah. So it's a solid source, I think, that is leaching into the dissolved phase and then being consumed rapidly. Okay. So to follow up on that question, we wanted to characterize what this source of organic carbon was. So we saw that there had to be some solid source that was supplying this, and we can now use the age to pin it down further and figure out what's going on here. What is this source? And now what I'm showing here on the right is the carbon age of the dissolved inorganic carbon, so the blue circles, the dissolved organic carbon, the red circles, and then sedimentary organic carbon and inorganic carbon. And again, don't get lost in the data, but just see that the dissolved inorganic carbon is much younger than all of the other sources of carbon that we've measured. Okay, so what does that say? It says that there's some young solid phase source of organic carbon that's required, because we knew that there had to be a solid phase source of organic carbon because our DIC generated was much greater than the dissolved organic carbon. And now we know it has to be quite young because what we've measured, at least, is much older. So there's something that we didn't measure that's very young. And we inferred that this DIC was being generated from the oxidation of pond muck. So in our pond, we have a lot of algae and plant matter settling every year at the bottom, and that can leach into the water as it recharges and be rapidly consumed. And to pin this down, so here's this slide showing, okay, this layer of pond muck. Now let's pin this down further, and we can do that by tracking the reactions that happen along a shallow flow path. So I'm showing here, let's take pond water. So this is surface water, and we're looking at DIC concentrations and DIC ages. And let's track that pond water going from the surface water down to 2-meter depth in the aquifer. In the aquifer directly beneath the pond. And there's a few things to point out. One is that we have a large increase. So if you look at that right column there, there's a large increase in the concentration of dissolved inorganic carbon. So there was a lot of oxidation of organic carbon. It generated DIC. Okay, and then we see that there's also a change in the dissolved inorganic carbon age along that flow path. So we know the age at the start and the end of the flow path, and we know the amount added. We can do some mass balance calculations and figure out the age of the organic carbon that was oxidized to produce this DIC. So we see that the DIC produced is, in fact, consistent with what I said on the prior slides. It's much younger than the dissolved organic carbon, the solid phase carbon as well. So we confirm there's this young reactive source of organic carbon that's in the solid phase that's generating the DIC and fueling these redox reactions beneath our pond. Okay, so we want to ask a few questions about this now, because we saw that we didn't have high arsenic beneath our pond, but we have very reactive organic carbon, and it's fueling redox reactions. So what is the question we can ask is, what is the age of labile organic carbon deeper in the aquifer where we do see high arsenic? So is it young surface organic carbon? So could it be that, organic carbon coming in from ponds and wetlands and rice fields is also carried to depth? It's, maybe it doesn't get fully consumed at the shallow zone. It gets carried to depth and continues to react and fuel these reactions. Or is it an old sedimentary source of organic carbon? So to look at that, I developed a set of laboratory experiments that further probe this question. So this experiment is carried out as follows. We go to a set of wells, and at each well, we collect samples in duplicate. One sample we take, we'd preserve it, and we'd immediately analyze the dissolved organic carbon age. Okay, then we take our duplicate sample, we'd bring it back to the lab, We'd inoculate it from microbes with microbes from that aquifer depth, and we'd let these reactions occur. So we'd let the microbes consume the dissolved organic carbon in this water sample. So remember, these were duplicates. We took one sample, measured the DOC age before any reactions happened, so we just pulled it out, pumped it from the aquifer, and another we allowed to react for some time, consume organic carbon, and then we took the age of that. Now by doing that, we can figure out the age of organic carbon that was consumed, right? The microbes are going to consume some of the organic carbon, and by getting the age at the end, we can see what the age change was, what the concentration change was, and through some mass balance calculations, figure out the age of the organic carbon that was consumed by the microbes. So we did that on 6 different locations. So we had 4 shallow locations, and those are the leftmost 4, and then we had 2 deep locations. So in the 4 shallow locations, we have all fairly low arsenic concentrations. In the 2 deep locations, we have high arsenic concentrations. The blue bars are showing the dissolved organic carbon age in those samples prior to the experiment. So we just pulled the sample out and immediately got the carbon age. And then the red bar adjacent to the blue bars is showing the post-experiment age. So it's that duplicate sample that we brought back to the lab, allowed microbes to eat some organic carbon, we'd see what they'd eat, and we took the age there. And you see here, now there's numbers above each of these sets of bars. This is the change in age. So something kind of interesting happened, and it was not what we were expecting. In all 6 of our samples, or all 6 of our experiments, the age went down. So we saw that we had the pre-experiment sample and the post-experiment sample, and in the post-experiment sample, the dissolved organic carbon age was younger at the end. So what does this mean? This actually means that old organic carbon was consumed preferentially, right? Because the remaining organic carbon, the stuff that didn't get consumed, was younger. So you ate the old stuff preferentially. So again, we can do some mass balance calculations, and we can actually calculate what the age of the organic carbon fraction that consumed, what the age of the organic fraction consumed was. And what we saw here in these shallow samples was that the age of the organic carbon consumed was around 3,000 to 3,500 years old. And what does this say? So this suggests that we've got reactive DOC in these shallow samples beneath our pond. And this reactive DOC, it's older than the bulk DOC. So it's older than the average age of the DOC, but it was younger than the sediment organic carbon. So this suggests that there's some kind of mixture of both sedimentary organic carbon and younger surface source carbon being consumed in these shallow samples. So the pond could have introduced some young reactive organic carbon. We saw very shallow that it was pond muck, and that got consumed and generated a lot of DIC. Then we go a few meters deeper, and we see that now the reactive organic carbon is older. It's some mixture between surface-derived and sediment-derived. Okay, so we're now curious. We've got some reactive organic carbon that made it a few meters in to the aquifer. Is that making it to 20 and 30 meters deep where we see high arsenic? So we had experiments that looked at the 20 and 30 meter depths where we observed high arsenic. And what we found here was that the organic carbon, the dissolved organic carbon that was consumed, was between 10 and 12,000 years old. Now these aquifers are Holocene in age, so they probably extend at most back to around 12,000, maybe a bit older. So this indicated that the reactive dissolved organic carbon is older than the bulk dissolved organic carbon. And in fact, the age of the organic carbon that was being consumed was, has an age signature that indicates sedimentary sourced. Okay, so this experiment suggests that we're seeing, sedimentary sourced organic carbon being consumed. But let's now do another kind of flow path calculation. So I did a flow path calculation for the shallow sample early. Let's do another flow path calculation going from 20 to 30 meters and see if we can find a result that's somewhat consistent with this. So from in situ generation of dissolved inorganic carbon. So we're doing that, and I'm showing here a plot of dissolved inorganic carbon versus depth. And what we're going to do now, like we did for the shallow samples where we went from the pond water to 2 meter depth, we're now going to track a sample from 20 meters to 30 meter depth. And what we want to see is what is the organic carbon that was oxidized, which generated this increase in dissolved inorganic carbon that we see. Okay, so here's a similar type of mass balance calculation. We see that there's about 3 millimolar of DIC is generated between 20 and 30 meters. So there was oxidation of organic carbon, and that would have fueled the redox reactions. And we saw a change in age. And what we found here was that the change in age was around, the change in age indicates that the organic carbon that was consumed along this flow path was around 4,000 years old. So this DIC that was input along this flow path is older than the DOC that exists on this flow path, but a bit younger, than the sediments. So now I want to put all these results together, both the in situ field samples as well as the laboratory samples, and let's see what we can say. So on this slide, I'm showing a summary of all my results here. On the left, you see a plot of arsenic with depth, just to put things in context. And on the right, you see the labile organic carbon. So this gray bar here has ages in it, and this is showing the age of the labile organic carbon, or the reactive organic carbon. So remember, we saw very, very shallow, in the upper few centimeters of the pond. So what we saw was that in the upper few meters of the pond, there was pond muck being consumed. So this was very reactive, surface-derived organic carbon. It was highly reactive. It generated the DIC. Then we went a little deeper in our experiments, and we saw, at a few meters beneath the pond, the labile organic carbon, definitely older than the pond muck, younger than the sediment. So it looked like it was some mixture. So there was some reactive, surface-derived organic carbon getting transported to depths of a few meters, and it was able to fuel reactions there. Now, when we go deeper into the aquifer, let's look at the two black numbers at depth, at 20 and 30 meter depth. Those are both my lab experiments. And what we saw in both lab experiments is that the labile organic carbon was almost, equal to the sediment age. So that suggested that at these depths where we see high arsenic, so you see that we have high arsenic at those depths, that this high arsenic is being mobilized through older organic carbon. That's what's fueling the reactions. And then we also did a flow path and mass balance calculation, and we to kind of see what was going on at that same depth. And that produced a slightly different result. It suggested that the DIC is being generated through the oxidation of both a mixture of sedimentary and surface-sourced organic carbon. So it's a little mixed there, but overall, it appeared that arsenic mobilization appears to be, mostly driven by sedimentary organic carbon. And I'll just highlight that we could do some kind of end-member calculations here, and it was a majority sediment sourced for this 4,000-year-old DIC generated, and a smaller portion, 30 or 40% of young pond muck. That's what it would come out to be. So it looks like, broadly speaking, it's driven by sedimentary organic carbon. Okay, so now we've got this result, and we think that sedimentary organic carbon is playing a really important role, with some secondary role played by surface sources here. But let's look at some additional geochemical evidence to try to pin this down. What we want to do is we want to pin down the source of organic carbon that's driving these reactions. We also want to try to explain the patterns of arsenic with depth and among a given depth. So we're going to look at a different type of geochemical measurement here. And I just want to remind you guys of what the arsenic concentrations with depth look like at this site. So we have arsenic increasing as you go down into the aquifer and you hit 30 meters. It reaches a really high level. But what you also see is that there's a wide range of variability in arsenic concentrations at any given depth. So let's take 30 meter depth, for instance. There's a huge range of arsenic concentrations here. So, what's causing that variability? Well, one hypothesis, and this has been put forward in the past to explain arsenic concentrations with depth, was that it might be the age of the water. So this is not invoking anything about the organic carbon source, just that the residence time of the water, as that increases, arsenic concentrations increase. And you can think about this simply, the longer the water is in contact with the sediment, the longer it has to accumulate arsenic in the dissolved phase. So, we took the groundwater age that was modeled for our field site at each depth, and I plotted that against the average arsenic concentration at each depth. And you see a pretty good linear relationship here, especially for those red circles, which is when the arsenic concentration is above the sediment sorption. So that indicates that there's a good relationship between arsenic and age here, and residence time might be playing a role. So we wanted to look at additional geochemical evidence, right? We don't have an age measurement for each sample that we've taken. Right? We only have average age modeled. But what I did was I used strontium as a proxy for relative groundwater age. And strontium can be released from the weathering of silicate minerals, and that weathering rate is relatively constant for a given pH. And over the pH ranges that we find in our aquifer, we expect that the weathering of silicate minerals to be reasonably constant. So if we take the strontium concentration of a sample, we can say, okay, that's a good indicator of the relative age of that water, how long it's been in contact with the sediment. And now I'm plotting all those samples, strontium versus arsenic here. And you see, especially for the samples above the sorption limit, we have a nice tight linear relationship here. So that indicates that arsenic is increasing linearly with age. That suggests that the variability in arsenic concentrations appears to be largely explained by differences in age. So remember at 30 meters, we saw a huge range in age in arsenic concentrations. Well, when we plot the arsenic concentration versus strontium, it just looks like the samples that had lower arsenic concentrations were just younger samples. They had made it to that depth faster and had been in contact with the sediments for less time. The samples at 30 meters that had really high arsenic concentrations were just a bit older, so that water had been in contact with the sediment for longer. So what does that imply? This has some interesting implications for perturbations to the system. Imagine if we have decreased flushing. So say we reduce the amount of irrigation pumping in our system, then at any given depth, the age is going to go up. So you have decreased flushing, and you can imagine taking this gray point and you'd move it upward along that line. At a given depth, it would be older, so the strontium would have increased, and so would the arsenic. Same thing here if we look at different thing here if we look at increased flushing. Imagine holding everything else constant, you just increase the flushing rate of the aquifer. Now at a given depth, the samples are younger, the water age is younger. It had less time in contact with the sediment, arsenic concentrations would go down. Okay, now these results also help us understand the organic carbon source driving these reactions. So imagine if it had been just surface-derived organic carbon that was responsible for these reactions. Well, then we would probably see a more decoupled relationship between arsenic concentrations and water age. Because you could have some really young samples, so the water recharged very recently, but it was rich in surface-derived organic carbon, and then it would have a really high arsenic concentration. So it wouldn't have a very high strontium concentration because it's fairly young, but the arsenic concentration would be elevated. Now, conversely, you could have a sample that's the same age as that high arsenic sample, but it didn't have any surface-derived organic carbon. So its strontium concentration is the same, but its arsenic concentration will be much lower because it didn't have, say, very reactive surface organic carbon in the recharge water. So that would have been a decoupling with the arsenic concentrations and age. So this suggests a more constant rate of arsenic release within our aquifer, which is more consistent of a sedimentary source of organic carbon. And so here we saw that the variability in arsenic concentrations is largely explained by variability in water age, and this strontium versus arsenic relationship helps to further support our contention that sedimentary organic carbon is playing a large role in driving arsenic mobilization. So now I just want to give you a summary of what we saw today and some concluding remarks. So we saw that redox processes at depth, they appear to be largely driven by sedimentary organic carbon, though there is a role of surface-derived organic carbon as well. And this helps us leads us to believe that high arsenic can occur absent reactive surface-derived organic carbon making it to depth. So high arsenic was potentially likely widespread prior to perturbations due to human beings. However, I really want to highlight that just because we see that sedimentary organic carbon may be the largest source of organic carbon driving these reactions, it does not rule out surface-derived organic carbon as playing a role at other sites or at other locations, or even at our site in the future. It just means at present we haven't seen a large influx of surface-derived organic carbon that's responsible for these reactions. Furthermore, we saw that arsenic is released at a relatively constant rate within our aquifer, and that this variability that we see both among depth and within a given depth can be largely explained by differences in the age of the water itself. And we saw that if we increase the flushing rate, we may decrease arsenic concentrations. If we decrease the flushing rate, we may increase arsenic concentrations. Further, and then finally, we saw beneath our study pond that at present we have low concentrations of arsenic. So we have not yet started to mobilize arsenic in large volumes there. And I didn't touch on this too much in this talk, but we at least hypothesize that there may be a limited ability for the pond to generate a plume of high arsenic. because we saw this pond was highly reactive, and it's possible that it will just be oxidized rapidly in the shallow sediments beneath the pond. So it might reduce all the iron oxides there, release some arsenic, but then once the iron oxides are depleted, if this reactive organic carbon is, say, consumed via methanogenesis and is not able to make it past this shallow depth, then it might not be carried very far into the aquifer, and it might confine the effect of the pond to a shallow zone. Yeah, and that's all. Thank you very much.
Some questions. Before that, though, I'll make a comment that maybe for those of you in public health, you didn't quite catch that. If you look at the past 10, 15 years, essentially, Charlie and the MIT group were big proponents of a surface-derived source of organic carbon driving this arsenic, and we can say, no, we don't think that's true. And what's interesting is that today, essentially, MIT says the opposite. Because of Brian Lee's work and Greg Seaton, we say actually surface-derived, so we're completely crossed in our opinion. We'll see how it evolves further. A couple of questions. Benjamin first, and then Jan.
So, what's actually the profile for the
Profile for the EH. So I mean, it gets more reducing with depth. I don't recall like specifically.
when you do the experiment, you do the surface.
Yes. Yeah.
Can you repeat the question?
Yeah.
So that
So he asked what the EH profile of the aquifer was, and then you were saying that when we did these experiments, we did it in the surface.
Yeah, in the surface and then in the air, in the
Yeah.
and so that kind of the experiment is showing that main representing the surface doesn't really representing these kind of the reactions happening in the subsurface.
That's a very good point. So kind of future work and next step is to do these type of reactions in a more representative environment. Yeah. So we would like to try that and see if we can we get the same exact result.
Okay, I think I'm still firmly in the sedimentary camp. One of the things that not know it because writing a paper since 2009 that was sedimentary. It was very nice to see that you went through the trouble to prove that it would be the case. And I have a question regarding how much do you have to force your model that the deeper parts have to have a surface-derived source, and you don't have to have it to make it work, right? In your deeper part, when your experiments show you only needed to have these older carbon because when you incubated it actually in younger. So that means that in your flow path you could allow for both the entry and surface derived. Do you have to have surface derived for your flow path to work?
Well, so the age of the organic carbon that was consumed along that flow path was around 4,100 years. So that is definitely younger than the sediment age. So it would indicate that there's some input. I mean, we don't. It could be shallower sediment. Yeah, that's true. Though I.
It's a tricky thing because sedimentary organic carbon has a wide spectrum of reactivity, and people now finding really old ancient permafrost is highly reactive.
Yeah, and so I'm definitely convinced that you can have highly active sedimentary carbon. And there's been Becca Newman did some experiments recently. She didn't date the carbon that was consumed, but just took like sediment cores from depth and then agitated them, and then saw a huge increase in like methane and DIC that was generated. So she was taking that was presumably old sedimentary organic carbon. By agitating them, it kicked it off the sediment, and maybe eliminated any physical protection mechanism that allowed that organic carbon to be preserved over time, and then entered into solution and was rapidly consumed. So I think that's kind of something that's happening here, is that we just have a slow leaching, and maybe the physical protection mechanism allows some old organic carbon to be conserved for long periods of time, even though it's reactive. But once it enters into solution, it can be consumed.
So I think it boils down to the iron oxide and not some of the implications there to show how it become reactive. And I think we're writing a paper now that shows it's a direct lovely mechanism, the electron shock wave, and what's limiting these diseases is really kinetic. Because the Unix needs to really turn the electrons around iron oxide to
Yeah.
If that's being so, follow it to look at it, really great, and then it's stacked.
Oh yeah, thank you. That's great points.
Someone else looking?
There's a question.
Oh yeah.
This is a question from Harshad Kulkarni at Kansas State University. How is the distribution of other electron acceptors, such as nitrate sulfates?
Mhm.
Is it possible that at shallower depths, there still may be some nitrate reducers that consume the organic carbon but did not respire iron-3 and released arsenic as compared to that at greater depths?
That's a great question. I actually had slides on that, and I took them out to make this fit better. But I was showing that beneath our pond, we had high sulfate. So this is similar to nitrate. You can have sulfate reducers. We had high sulfate when we installed the pond, and that's one thing we've really seen change. That's been dropping over time. So that could be one of the reasons why we're not seeing arsenic mobilization beneath our pond, is that maybe it's being, the oxidation of organic carbon is being coupled to the reduction of sulfate. Or simultaneously iron oxide reduction and sulfate reduction, but then that could precipitate as sulfides and scavenge out with arsenic. Yes, that's a really great question, and I think that's at least part of the reason why we might not see arsenic mobilization beneath our pond right